Hi, my name is Steven Kuzner, and I'm a sophomore majoring in environmental microbiology. I do my research here at WVU under Dr. Ember Morrissey in the Division of Plant and Soil Sciences. So today I'm presenting my research, which is titled Assessing the Impacts of Hydraulic Fracturing on Stream Health Using Algal Diversity and Biofilms. So hydraulic fracturing, which is commonly known as fracking, is a method of extracting unconventional oil and gas that involves injecting high-pressure liquids into bedrock. This causes a fracture and releases natural gas. So the mismanagement of wastewater at fracking wells can lead to ground and surface water pollution, which has the potential to impact streams. Land disturbance and deforestation associated with well pads and pipelines may alter sediment delivery, change nutrient levels, and affect light availability in streams. Here in the diagram, I show a little bit about the process of hydraulic fracturing. And you can see that through the process of causing a fracture, there is potential for groundwater contamination if there's like groundwater in the general area of the fracking. So stream biofilms are layers of microorganisms such as bacteria and eukaryotic algae that adhere to benthic surfaces. Biofilms are the base of the food web, meaning that any environmental disturbances that alter these biofilm communities have the potential to impact all trophic levels. So the goal and hypothesis of this project. So our goal was to determine the effects of hydraulic fracturing on eukaryotic algae within stream biofilms. Our hypothesis was that hydraulic fracturing activity within a watershed affects stream biofilms, thus altering the proportion of algae and changing algal diversity. So to determine this, we first collected biofilm, soil, and water samples within 26 streams in the state of West Virginia. So there were 19 impacted streams and 7 streams that were used as reference. So these streams do not have any hydraulic fracturing in the watershed. So the level of impactment was determined by the acres of pipeline, the number of wells, and the area of oil and gas land use in each watershed. From this, we determined 3 levels of impactment: non-impacted, which serves as a reference, impacted, and highly impacted. For each stream, the water and sediment pH, electrical conductivity, and oxidation reduction potential were recorded. DNA was extracted from the biofilm samples. Next, polymerase chain reactions, or PCR, was used to target eukaryotic organisms through the 18S rRNA gene. And finally, samples were sequenced for the 18S rRNA gene. So in terms of our results, we have 2 different sort of sections of results, and that's algal diversity and then algal abundance. So within algal diversity, these 2 figures show the variation in biofilm algal diversity, which we use as Shannon Diversity Index, and that's the figure on the left, which is A. And for the beta diversity, we analyze this using the Bray-Curtis dissimilarity model, which is on the right. So these varied across streams with varying levels of hydraulic fracturing activity within the watershed. So we determined that algal alpha diversity, which is indicative of species richness, was similar across all impactment levels, meaning that there wasn't really a difference depending on if there was hydraulic fracturing in the watershed or not. So algal beta diversity, however, which is the figure on the right, which is indicative of community composition, was less variable in the highly impacted sites. This suggests that hydraulic fracturing activity may have a homogenizing effect on algal communities. So in terms of algal abundance, we determined this through the relative abundance of photoautotrophs, or algae, based on the percent of the algae in the 18 rRNS genes. So we found that impacted sites had a higher proportion of algae in biofilm communities. So we interpret this to mean that the increase in algal abundance is most likely due to more light availability associated with deforestation in the watershed. This can mean that streams receive fewer litter inputs and have more algal productivity. This may change the food web because these invertebrates feed on litter leaf and algal biofilms. Changes to those invertebrates will directly impact fish populations, which has the propensity to impact all trophic levels. Our hypothesis is supported by our findings and thus supports the idea that hydraulic fracturing within a watershed affects algal diversity and presence. These findings may help to contribute to a better understanding of how unconventional oil and gas development affects microbial communities and broader ecosystem functioning. So this study was made possible by a National Science Foundation EBSCO award that was entitled Improving Water Management, Treatment, and Recovery in Oil and Gas Production. So that concludes my presentation, but if you guys have any questions, please feel free to reach out in the comments. Thank you.